Go. Well, welcome everybody to Pushing the Limits. This week I have a very special interview continuing our series around intravenous vitamin C, or vitamin C in general. I have Dr. Berry Fowler with me, who is sitting in Virginia, and Dr. Fowler has agreed to come and have a little chat today about his work in this area. Dr. Fowler, I've done a wonderful extra introduction, so we won't go into all your amazing credentials and your achievements, of which there have been many. But Dr. Fowler, can you just give us a little bit of background? You are the director of the VCU at Virginia University over in the States. Can you tell us a little bit about your work and your background?
Okay. Well, I am professor of medicine in the Division of Pulmonary Disease and Critical Care Medicine. And I'm one of the ancient doctors in the division, just turning 71 last week.
Congrats.
I trained at the Medical College of Georgia in the US, then went to the Medical College of Virginia in the US, then went to the University of Colorado for pulmonary and critical care disease training, and then came back and joined the faculty at Virginia Commonwealth University, which used to be the Medical College of Virginia. It's now VCU, in 1982, and I've been here ever since.
Wow.
I rose slowly through the ranks. I led the pulmonary division for a number of years, for approximately 17 years, and then stepped aside in 2016. And all during this time, at least for 13 years now, we've had this interest in vitamin C. And it's interesting how our interest in vitamin C developed. It first started at a very molecular level, where we were studying cardiac ischemia with some of the heart attendings, and then slowly began to get back to what we had been doing for years, which was bacterial sepsis. And we had some molecular reasons that drove us towards vitamin C. And so first thing we did was we created an animal model of sepsis. And let me explain that. It was pretty straightforward to create. We had 30-gram mice, and we went to the mouse cage and collected mouse pellets, then took them into the laboratory and sonicated them really hard until it became a solution.
So this is the fecal matter.
And we would take that solution and centrifuge it really hard so that all the solid matter went to the bottom of the tube, and we just took off the liquid from the top, which contained multiple different kinds of organisms.
So all the bacteria.
Yes. And so we took that, put it in the refrigerator overnight, and then came in the next morning. And we had 10 mice. We had 5 control mice, and then 5 treatment mice. So all the mice first were injected into their peritoneal space, you mentioned that earlier, with 1/10 of an ml of this solution containing all this bacteria.
Wow.
And so all 10 mice. And then in the mice that were going to receive the vitamin C, we injected 1/10 of an ml, which was 200 micrograms per gram of body weight of the mice. And then closed off the light. By that point, it was about 4 o'clock in the afternoon, and just let the mice sit in the laboratory where we had left them. And I always get to work at 6 in the morning, and I was thinking, holy cow, I got to see what's going on. And so I went into the lab where we had the mice, and in the cage that was the control mice that were septic, they were all dead.
All dead. So
in the cage that the mice got the sepsis and the vitamin C, they were all crawling around, drinking water and eating.
Wow.
And I knew at that point that we had stumbled on something pretty significant. This takes us back to around 2010, maybe 2009. And My laboratory has had this intense interest in sepsis ever since I finished my training at the University of Colorado. And so what we decided is that we would begin to use the treatment animals and some control animals to determine exactly how vitamin C was working.
So to look at the mechanism of action, why is this happening? Why are they surviving better?
So what we did was we in these studies, we were always comparing the control mice to the treated mice. And one of the first things we found was that the lungs of the treated mice that were septic, they weren't injured.
Wow.
And we have a number of ways to determine the way a lung is injured. One of the things that happens in sepsis, and this might have been what you and I were talking about earlier, is the lung's barrier function, which is the ability to keep the blood in the blood and keep the air in the air.
Yep.
It gets injured. And so the bloodstream floods into the air spaces of the lung.
And fills it.
Yep. And one of the things we discovered was lung barrier function was preserved in the vitamin C treated septic mice.
Wow. So you're preserving that, you're stopping the plasma and the neutrophils getting into the actually alveolar space.
Exactly.
And the nets, I've heard one of your lectures, you talk about neutrophilic extracellular traps. Is that a part of that barrier function?
Very nice. When are you starting medical school? Remind me.
Thank you, Dr. Fowler.
So what happens as sepsis progresses is that there are a bunch of molecules that live in the capillaries of the lung that begin to get expressed.
Yeah.
And what they do is they trap neutrophils that are activated in the capillary space of the lung. And one of the things that happens in a highly activated neutrophil is they disgorge their DNA.
The DNA.
And all of the enzyme systems inside a neutrophil begin to damage the capillaries. And then what happens as the capillaries get injured, the plasma from the lung from the bloodstream just flows into the
pours in. So you're basically lost. It's like your skin barrier, if you like, between the air and your insides is disintegrating.
Well, lung injury from sepsis is like drowning.
Wow, so you're filling with your own fluid.
The air spaces of the lung fill up with your own plasma.
So when you have, because sepsis, I don't think most people are not aware of the progression of sepsis to ARDS, so acute respiratory distress syndrome, that this is a sort of a linear progression that happens, isn't it? That you actually get lung, because most people understand sepsis as being a bacterial infection, but they don't understand that it's actually attacking the organs and causing oxidative damage to multiple organs, not just the lungs, but particularly the lungs. And so this is a very important finding that what you've had here, because this means that if you can stop the vitamin C, if the vitamin C can stop the neutrophils from disgorging their own DNA into the extracellular space, which is then, that's a marker, isn't it? That cell-free DNA, when you take a plasma draw and you see that cell-free DNA floating around at a certain level, that's a predictor of mortality, isn't it? So it's a very important mechanism.
Well, listen, you've done some fabulous reading, but let me just tell you, it's been known for several years that in septic individuals, one of the unfortunate things that will predict mortality is how high the cell-free DNA arises in the circulation. And I don't want to jump too far ahead, but I will tell you, in the vitamin C trial that we reported 1 year ago this month, that when we reanalyzed the blood from those individuals, we found that vitamin C dramatically lowered the cell-free DNA in the treated patients.
Oh, wow. That was in the CITRIS-ALI study?
Exactly.
Oh, okay. That's a new finding from that study, because we will go through that progression of how you got to do that study. So let's bookmark that for a moment and backtrack, because that is a very important finding then for that study, because that so let's backtrack a little bit. So we're talking about vitamin C being able to protect the lungs, if we put it very simply, and protect the barrier function of the lungs, stop the neutrophils from disgorging their DNA and causing these traps, which is a predictor of mortality. What other things is vitamin C doing, and why is a septic patient without fail going to be very low in vitamin C? So you're using up your vitamin C.
I'll get to that in a minute. But what we demonstrated in a huge number of murine mouse studies is that the septic lung in a control animal, the septic lung began to express many inflammatory proteins. Mm-hmm. And that's just your endogenous immune system trying to protect itself. But we showed in the next cage, in the septic mice that we had treated with vitamin C, that the expression and the appearance of those inflammatory proteins was totally inhibited. Wow. Completely.
Wow.
Yeah.
Okay.
So the idea of leaping from preclinical animal studies into humans was that we had kind of a basic grasp on the immune system and how vitamin C could alter the septic immune response, mm-hmm. and how vitamin C could protect the lung.
Right.
And
so
protecting the lung.
Yep.
Well, protecting the lung in terms of septic critical illness is very, very important. Absolutely.
And so then you went to a phase 1 safety trial, which was really to look at the some basic markers. Is this going to be damaging for people if they get vitamin C, and looking at hypertension and is it going to affect the kidneys and so on? I think some of those safety mechanisms. Can you tell us a little bit about that phase 1 safety trial and then the outcomes of that trial?
Well, I can tell you, I had this really close colleague his office sat right next to mine. He's a molecular biologist, basic scientist. And after we'd done all these murine studies, one day he walked in, he looked at me, he said, "Fowler, this needs to go into the hospital. We've developed all this data. You've got to make it happen to get it into the hospital."
Dr. Ramesh.
So we designed this little safety trial, enrolled 24 patients. The safety trial was randomized and it was blinded. Mm-hmm. And so half the trial was just control sepsis. The other half was septic patients treated with vitamin C. And we had no idea who the hell was giving vitamin C to people who were critically ill.
Yeah.
And we found that it had no impact. But one of the things we were shocked at, and we were just trying to define, was vitamin C safe?
Yes.
One of the things we tracked was what is called an organ failure score. Mm-hmm. And we found that all of the patients treated with vitamin C, their organ failure score reduced dramatically and significantly.
Wow.
And the way organ failure scores, you basically you're counting numbers. A higher number is higher incidence of mortality. Mm-hmm. Lower numbers are improved, and that vitamin C was actually improving the possibility of survival.
So this is like in my father's case, as the sepsis progressed and I was unable to get him vitamin C, as we discussed earlier, Dr. Fowler, early enough for him to get to survive. But as I watched his sepsis progress, more and more organs started to fail. So his liver started to fail, his kidneys started to fail, his heart started to fail. And so this is the organ failure score. So if a person's organ failure score is going up, that is a very strong predictor of mortality.
Yes.
Okay, so this was reduced with the people who received the vitamin C in this small trial.
So what we did, we took the data, we combined it with our preclinical data and applied to the National Heart, Lung, and Blood Institute. They had just published an announcement where they were asking for anybody who could think of some clever trial.
Mm-hmm.
And we said, well, and so we submitted an application. What the NIH wanted, they wanted the proposal for a phase 2 proof of concept trial.
Right.
And so what we proposed was a trial that had 7 medical centers. I had friends in 7 medical centers around the US, and put this application in. And that was, I guess you guys don't remember Hurricane Sandy.
Yes, we do.
Hurricane Sandy was just, it killed the Atlantic coast of the US. And the National Heart, Lung, and Blood Institute happens to sit on the Atlantic coast in Washington, D.C.
Mm-hmm.
And it was a year and a half before we found out that we had received the highest priority score because of the application that we had submitted.
Brilliant.
And the NIH gave us $3.2 million to do a multi-center, randomized, double-blind, placebo-controlled trial, proposing to administer 50 milligrams per kilogram of intravenous vitamin C every 6 hours for 96 hours.
Mm-hmm.
So patients were continuously receiving vitamin C.
Can you explain why that continuous topping up that level is important every 6 hours?
Oh, that's a great question. So from the safety trial that we had performed, we analyzed the vitamin C, the plasma vitamin C levels that we had achieved by infusing. So basically, someone your size, for example, would probably get maybe 3.5 grams intravenously every 6 hours for 96 hours.
Mm-hmm.
And what we showed was we could get the plasma level up to basically 3,000 times the normal plasma level.
Wow.
So from a normal diet, human plasma levels of vitamin C are about 70 to 80 micromolar.
Yep.
When you give the protocol that we had settled with, we got the vitamin C levels up to 5 millimolar.
Wow.
Yeah. And so that's what we were shooting for in this NIH trial. And that's what we did. We charged into it, the trial. What we had proposed, again, was the organ failure score, as well as the 2 biomarkers. We also proposed in the secondary outcomes, days on mechanical ventilation.
Yeah, which is hugely important.
And what we were studying specifically was patients who were septic, who had gone on to develop acute lung injury called acute respiratory distress syndrome, ARDS.
Yep.
And so when a patient was septic like your father, we would become a fly on the wall and visit the patient every day until lung injury developed. And that's when they would get randomized.
This was a critical for my analysis of the data. That was a critical thing in the piece. So you had to wait until they basically had developed ARDS before you were able to put them. So this wasn't really a sepsis trial, but more of an ARDS trial. So the progression of the sickness comes into play here, doesn't it? If you've got it from day one, like in the phase.
In the safety trial.
Yeah.
The second a septic individual walked in the door, that's when they got randomized.
Which is a much better, more, effective with the timing.
We had a couple of patients who got vitamin C in the emergency room.
Yeah, wow.
Or, you have to get informed consent. You have to get the pharmacy on board and get the patient infused.
Oh, I wish I'd had you attending to my father, and we could have had that from the moment he got to the emergency. That would have been, I think we would have had a different outcome. But so this was very a key point, that you had to wait until they had developed ARDS So in this CITRIS-ALI trial, so here you have, I think it was 47 patients in the control and 47 in the intervention group. Was that right?
83.
Oh, 83. Sorry, that was
84
Dr.
in the
Merrick's.
vitamin C treated group.
Yep. So 167. Why was, like, one of the big questions I had in my thing was, why was mortality not one of the primary objectives of this study?
That has been the most frequent question. When we answered the NIH, they had put out a program called UM1, and that's who we applied. We applied to the UM1 program, and they were not interested in mortality as a primary outcome. Part of it was this: there had been hundreds of sepsis trials, and nobody had ever shown any impact on a treatment for sepsis.
Gotcha.
And so NIH didn't want to get burned again. So they said that they wanted a physiological outcome. That was the organ failure score. And they wanted a biochemical outcome. Those were the biomarkers.
That's your C-reactive protein, your procalcinin, thrombomodulin, and yeah. So the reasoning was that we don't want to shoot for the stars here and automatically hope for a decrease in mortality and a decrease of days in hospital. We're going to go for something less just to see if this has legs, so to speak, if this treatment is possibly going to work. And that's why they went for the SOFA scores and the rather than the mortality. Looking back, do you think?
And by the way, we haven't talked about this yet, but SOFA stands for Sequential Organ Failure Assessment Score.
Thank you. Yeah, it's amazing the jargon that you pick up and then forget that you haven't explained yourself. So what actually was the outcome? This was a 7-center, multi-center trial. You did double-blinded. This was incredibly important because I know Dr. Paul Marek had also done a study with intravenous vitamin C, thiamine, and hydrocortisone. And one of the criticisms that was thrown at him was that it wasn't a double-blinded randomized control trial, so it didn't have any meaning, which is absolutely tragic. So this was. What was the outcomes of this phase 2 trial?
So we enrolled 170 patients. One of the placebo patients we had to take out because that patient did not have septic ARDS. They had acute eosinophilic pneumonia. That's something else to discuss later. And then in the vitamin C arm, we had 2 patients with acute leukemia who had no coagulation in their bloodstream, and they were hemorrhaging into their lung, and that was not sepsis.
Yeah, it's another thing.
So as I mentioned, we had 83 in the control, placebo, and 84 in the vitamin C treated group. First of all, we saw no, and I emphasize capital NO, adverse events. There was not a single adverse event.
Exactly.
All right. And so what we showed was in 96 hours, placebo patients in the trial, 19 of 83 died within 96 hours.
Wow.
In the vitamin C group, 4 of 84 patients died. And if you look at the statistics and the analysis of that, the difference is P equals 0.0007. We then followed the patients out because in sepsis trials, there's always this demand to see what is happening to a patient at 28 days.
Yes, yeah.
And what we showed was 46% of placebo patients died, and only 30% of the vitamin C treated septic patients with ARDS died.
Wow, that's a huge result in my mind.
Yeah, that was the first trial. I'm not slapping myself on the back, but I will just tell you, that was the first trial to ever show in a blinded fashion an impact on ARDS.
Yeah, on mortality of ARDS.
Yes.
And this was extremely sick people. Now, unfortunately, the SOFA scores didn't show any difference, and the C-reactive protein markers didn't show any difference.
So let me explain.
Because, is
Yeah,
that
hold on.
because of the mortality?
So we thought publishing the results of the trial in probably one of the most important journals on the planet, JAMA, which, as it turns out, is a very, very conservative journal. And they had their ideas about what we could and we couldn't say. So we published, and this is very important for you to listen to and all of your listeners. We published that there was no difference in the SOFA scores at 96 hours. And immediately letters to the editor started coming in. And one of the most important letters to the editor was the person who created the SOFA score. His name is Jean-Louis Vincent in Brussels, Belgium. He told us that we had analyzed the data incorrectly and that what we were reporting was a survivorship bias.
What does that mean?
And what he said we needed to do, and he provided 5 publications where he had important statisticians tell him that analyzing the data like we reported as a worst rank, best rank scenario, that we had to reanalyze it so that the patients who died, what we were reporting was the SOFA scores on the people who had survived.
We had not
considered the SOFA score on the patients who died.
And because they died so quickly.
So what we did was we went back, and the people who died along the way, those 19 patients, they got the top SOFA score. The patients who survived and left the unit, they got a low SOFA score. And so when we reanalyzed the data, according to the way these letters that had come in from Dr. Vincent and 2 or 3 other colleagues, it turns out that vitamin C significantly impacted the organ failure score.
Wow.
And then here's the important thing, we reported that February 25th of 2020. So you can go to JAMA, you can look it up, and you can see our response to the SOFA score reanalysis.
Because this was a key factor in my father's case. They threw the CITRIS-ALI trial at me and the original data from JAMA, which said negative result, which that
lets you know that the doctors were not reading JAMA.
Exactly, and they weren't on the up-to-date, and they did not look at secondary outcomes, and they did not look at the parameters of the score, and I was not able to present the case. They had just read it briefly.
Let me go on. We had a strong trend to ventilator-free days in the people who got the vitamin C, but it just missed statistical significance.
Yeah.
But we had a strong significance for the people who got vitamin C in intensive care unit free days.
Which is huge.
So the people who got vitamin C had significantly higher number of ICU free days. There was an improved mortality. And the other thing is patients who got vitamin C had significantly more hospital free days at day 60.
Wow. So they were actually out of the system altogether. Do you think, now this is a controversial, I'm playing devil's advocate here, but do you think the fact that it costs so much for someone to be in ICU when they have sepsis, and I think in America it's something like to the order of $60,000 US a day, and the medications that they are typically on are costing around $20,000 a day. Do you think that if you come along with vitamin C and you start dropping the mortality rate, you start dropping the days, is that part of the resistance to accept and acknowledge these findings, that the pharmaceutical companies are going to lose out on profit?
Oh, no. At VCU, Virginia Commonwealth University, that Anitra knows well, the average care cost per day is about $46,000 per day, because that accounts for medical care, nursing care, radiology, all laboratory data, respiratory care, caring for the ventilator. All of that is somewhere in the neighborhood of about $45,000 to $50,000 per day. And so if you have a treatment, first of all, that gets people out of the ICU earlier and keeps them out of the hospital, think about the impact on the cost of care.
Yeah, well, it'd be
Believe me,
huge.
but here's the other thing. There's not going to be any drug company out there who would argue with that. They're all trying to do the best they can with their different antibiotics, but the common antibiotics that are administered in an ICU when patients are septic, levofloxacin, meropenem, vancomycin, just one day of meropenem is $1,500 per day.
Exactly. It's a lot of money.
Yeah.
So, so you don't think that that's
listen to
influencing?
This. That's the cost of the drug. That's not the cost of pharmacy preparing the drug, of cost of nursing administering the drug, and so on and so on and so on.
Okay. So, all right. So we're gonna, if you can work this problem out, and if you can get this in all ICUs around the world, we're going to save not only thousands and eventually even more, hundreds of thousands of lives. You're going to reduce the hospital bills enormously. So this is incredibly important work. So, and you've proven, so the statisticians proved in that phase 2 trial that the way that you were measuring it was incorrect, because a lot of people, as you said, 19 died in those first 4 days in the control group and only 4. So that skewed, if you like, the statistics to initially look like we hadn't had a win here. Now that's been rescinded, and you've managed to get JAMA to publish it in a different light, that the SOFA score was impacted. What has been the effect now? Have you got another trial underway, or have you got one in sight? Because this work's too important, obviously, not to be taken further into a phase 3.
All right, so you are in New Zealand, where there's not much COVID.
No.
We are in the United States, where it's a pandemic, where we are close to 220,000 people who have died from the virus. We are at 50,000 new cases per day.
Oh my God, that's so.
And there are somewhere in the neighborhood of 1,800 to 2,000 patients dying per day of COVID. And so because of that, the network that I'm part of, that unfortunately I'm going to have to jump off and listen to because it's been going on since 2 o'clock, the annual meeting of the Prevention and Early Treatment of Acute Lung Injury Network, abbreviated
PETAL.
PETAL, the PETAL network. The PETAL network was tasked by the NIH to turn sharply towards COVID treatments.
That makes sense.
And so we were thinking, well, maybe vitamin C to treat patients with early COVID pneumonia. And so what we did was we started a trial. We have studied 20 patients now, and that trial is complete.
Wow.
Where patients who develop COVID infection and develop early COVID pneumonia, so it's just at the start of an oxygen requirement, are treated with vitamin C, and the results have been pretty dramatic. Wow. We are in the midst of writing that up, but again, it's a open label trial. It's not blinded. Everybody in the world knows that an open label trial has not have the power like we did with CITRIS-ALI.
Yeah.
And so what is happening at a world level is that all of the health organizations around the world have come to bear to try to design treatments for COVID pneumonia.
Yeah. And
that is ongoing right now. And there are like 9 or 10 major networks across the world. Probably, I'm not sure if New Zealand is included in that, but Europe, the US, possibly Australia. I don't know if they're participating in what is called the network of networks formation.
Yep.
So right now, the next trial for patients with sepsis that's not COVID is going to be conducted by the PETAL network, where we will be probably next April starting a trial with 1,000 patients.
Wow.
using vitamin C, conducted by the PETAL network.
Gotcha.
And the PETAL network has 69 medical centers. So doing a trial that would get 1,000 patients can be done within a year.
Wow. So this is exciting stuff, because this is hopefully you'll be able to reproduce and show a strong correlation between intravenous vitamin C and, well, I'd like to see, the decrease in the mortality rate. That would be a key factor. Some centers are already using vitamin C because, as you mentioned before, there were no adverse reactions. And this is like in all of the studies that I've seen, there has never, this is a low risk intervention. And my argument when fighting for my father was that he's dying, there is no other options. Why can't I throw the bus at it? Why can't I put intravenous vitamin C? And they were like, you still have to go through all the ethics committees. I had to get sign off from every single doctor and every single nurse in that ICU unit, of which there are many.
Well, let me make another statement. Paul Merrick, who was using 1.5 grams of vitamin C, 200 milligrams of thiamine, and 50 milligrams of hydrocortisone administered every 6 hours. That meant that the patients were only getting 7 grams.
Yeah, very small amount.
In the CITRIS-ALI, I mean, some patients got 16 to 18 or 20 grams.
Yeah.
According to body weights, 50 milligrams per kilogram.
Mm-hmm.
In the aftermath of that article that you mentioned that Merrick published, there have been efforts to recapitulate that trial.
Yeah.
The vitamins trial came out in January using that, and it failed. Then another trial, the ACTS trial using the Merrick protocol, failed. And then a trial that I just participated in called the Victus trial.
Yes, I'm aware of that one.
Completely failed. And so the Merrick protocol is not an effective treatment for sepsis.
Is it too low a dose?
Well, look, as I think Anitra Carr mentioned to me a couple of years back, the amount of vitamin C that you administer is critical.
Absolutely.
So dose matters.
Yes.
And the adult, again, of your size, you probably weigh 120 pounds or something,
Yep,
would probably get would probably get somewhere in the neighborhood of about 12 and a half to 13 grams spread out over a 24-hour period, and then you would get it for 4 days.
Yeah, and that is still a relatively low dose.
It is.
When I'm doing intravenous vitamin C with my mum, I did it with my dad prior, unfortunately months prior to his aneurysm. Too little, too late. We were getting 30 grams. We get 30 grams a week when I take my mum in yesterday for an intravenous vitamin C as a prophylactic, as a to try and keep her as a 79-year-old healthy. 30 grams. So why I had this question, certainly with Dr. Merrick's protocol, it seemed to me to be very low, although the 6-hourly is obviously a very important point as well. Why not do the bigger dosages? Like in Japan, I know they did a study with up to 100 grams of vitamin C in a burns case, a burns trial, where they had some marked success there. Why are you not trying higher levels?
Let me come in here quick, because I'm going to have to jump off in about 8 minutes. But listen to this. The major concern were those high doses of vitamin C. And if you talk to the oncologists who have been using it for years, they will give, like you said, they will give massive doses.
Yeah.
And I'm talking massive, like in somebody with pancreatic cancer, they will get 60 to 80 grams intravenously Monday, Wednesday, and Friday for 7 weeks.
Yeah.
But the major concern in somebody who's septic, who's hypotensive and shock, that you're giving vitamin C, one of the major concerns is that it causes a significant rise in oxalate crystal formation in the kidneys. Now, I will mention here, in the CITRIS trial, we had no evidence of renal stone formation.
No. And I mean, that was one of the arguments that the doctors had at me. You could damage his kidneys. And I said, well, the last time I looked, being dead damages your kidneys too. You know, like, to me, that wasn't even a consideration. And he had, after the very first vitamin C for my dad, his kidney function went from 27% to 33%. He's actually improved his kidney function overnight. And I know that's just one anecdotal case, but kidney stones are not going to kill you either. So surely that's not the most important consideration here when you've got a septic patient who is on death's doorstep.
Well, with vitamin C struggling in the United States after the CITRIS trial, the Federal Food and Drug Administration, they always have to be concerned about adverse events. And we have put together a trial, randomized and double-blind, using vitamin C in patients with COVID pneumonia. That's about to start.
Wonderful.
And I unfortunately let my IND, investigational new drug, lapse after CITRIS. And so I've had to claw our way back into the good graces of the FDA. And one of their major complaints was, you're going to be forming renal stones. And we're using the same protocol that we used in CITRIS. So FDA got their nephrologists involved and finally gave us the IND. But for us to begin treatment of COVID pneumonia, they have demanded that we first do a small safety trial to show that we are not causing any renal stone formation.
And how many people are dying?
Look, we can get that done. We currently have somewhere in the neighborhood of 60 to 70 COVID patients in the MCV hospitals right now.
Wow. Well, Dr. Fowler, look, I know I'd love to have spent another 5 hours with you discussing all this, because I think it's incredibly important work, both for COVID and for the sepsis and for pneumonia and for, obviously, for cancer. I just want to thank you for your dedication to this. I mean, you could be in retirement and sunning yourself somewhere and relaxing, but you know
that's
that
right.
You know that this work is critically important. And I heard in one of your lectures, there's the equivalent of 2 747 planes going down every day filled with people
in the United States.
In the United States
That's just
alone.
in the US.
Yeah. And these are people, thousands of families being destroyed with losing loved ones. I'm one of those, unfortunately, sitting here all the way in New Zealand. And so this work is incredibly important. So please keep going. And I'm desperate to hear what comes from this COVID clinical trials and the other sepsis trials, obviously. So thank you so much for your work, Dr. Fowler, and I really appreciate you.
It's been wonderful meeting you and speaking with you and your audience. And when you have Anitra on in a couple of weeks, give her my regards.
I will definitely do that, Dr. Fowler. That's been awesome. Thank you, Dr. Fowler, and all the very best to you in Virginia.
Take care. Bye-bye.